Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science Podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science Podcast. I'm Ayesha Rashid, Senior Life Science Journalist at Xtalks.com, and this week I am so happy to introduce a special guest on our show today. We have with us Dr. Edwin Tucker, who's the Chief Medical Officer at Jasper Therapeutics, which is a company focused on targeting mast cells to treat chronic mast cell diseases like asthma, food allergies, and IBS, among others. Dr. Tucker has over 30 years of clinical experience leading novel drug development. He most recently served as Chief Medical Officer at Goldfinch Bio, where he led clinical development and helped build the regulatory, medical affairs, and clinical operations functions. Dr. Tucker previously served as Chief Medical Officer at Mirum Pharmaceuticals, helping achieve key milestones, including the first FDA approval for Alagille Syndrome. Before that, he was Chief Operating Officer at Acerta Pharma, which is now part of AstraZeneca. And he's also held leadership roles in development, medical safety, and medical affairs at Genentech, Janssen and Bayer. Dr. Tucker is a member of the Royal College of Physicians UK. He received his MBA from the University of Connecticut. Dr. Tucker holds degrees in Pharmacology and Medicine from the University of Leeds in the UK. And in addition, he serves as a managing director at Golden Seeds, which is an investment firm dedicated to pursuing early-stage investment opportunities in women-led businesses. To learn more about targeting mast cell diseases and the work that Dr. Tucker is leading in this space at Jasper Therapeutics, let's turn to him now. Thank you so much for being on the show with us today, Dr. Tucker.
Thank you, Ayesha. It's great to be here. Thank you very much for that nice, warm welcome. I'm excited to be on the podcast today.
Oh, likewise. We're so excited to have you and to learn all about, again, like I said, the work that you're leading in this space, mast cell diseases, which you don't hear too much about. So I'm personally very excited to learn more as well. And so I'd just like to start off by talking more about, your fascinating career in drug development and the pharmaceutical industry. And I'd like to know what really initially drew you to drug development from medicine, from clinical practice, and what led you ultimately now to join Jasper Therapeutics.
Well, thank you. It's been a longer career than actually you detailed. My first foray into the industry was in the late 80s. Actually predated my start in my studies in medical school in Leeds. And so that gave me an early insight into the industry. And during both my studies and also my practice, I maintained both an interest and also an engagement with the industry. So whilst training in internal medicine and cardiology, I was also involved with several large pharmaceutical companies around emerging assets that were coming through in the cardiovascular space. At the time, it was the ACE inhibitors. And that was really my entree into Bayer, which was my first company after leaving clinical medicine and academic cardiology in Manchester, in England, that I began running clinical trials for Bayer in fairly large studies in a statin study that was run from the Oxford Centre in the UK, and this was part of a larger global program. But as many of your listeners might know, rosuvastatin was withdrawn from the market about a year after I began at Bayer. So there was a very interesting low point in my career, seeing my product being withdrawn for safety reasons. I continued to have, I think, a very interesting career with Bayer, and I still covered my time with Bayer very warmly. It was a great training ground for me as a relatively new person coming into the industry. And had experiences across large molecules with the biologics business we had in Berkeley when I moved to the United States, and then moving back into the small molecule assets on the East Coast with Bayer Pharmaceuticals. And then finally, when we integrated our first acquisition, which was Schering AG, into the Bayer company, we then had experiences across a number of different molecular sizes, both antibody and small molecule, and also blood-derived products as well. And so those were great years, 10 years at Bayer. And then I moved to Johnson & Johnson, again, a large pharma business out on the East Coast, but really having that global presence as well. And that gave me additional facets to my career. And then I moved back to the West Coast to Genentech, a little bit closer to the science. And obviously, a lot of experience generated at Genentech. And I was fortunate to be there for about a year and a half at Genentech before moving into small startup biotech. And as you mentioned, I began at Acerta, was my first biotech company in the area, a stealth company that was not long after I joined. It became part of the AstraZeneca family of companies as we began to advance acalabrutinib, which was the second BTK to market. And we were fortunate enough to achieve an indication there of mantle cell lymphoma, and that drug has just done incredibly well to help patients with hematological malignancies.
Wow, fascinating journey indeed. And thank you so much for sharing all of those details around it. And it's, it must have been quite the transition going from not only academia to industry, but you had that transition quite early on, as you mentioned, while you were still in medical school, but really from going from big pharma to smaller biotechs. And that seems to be really where a lot of the innovation is being driven, dare I say, in the smaller biotechs, not to knock on the big, pharma players at all. But how has that transition been, and what have the key differences and similarities been as well?
Yeah, so I do think there's a lot of amazing innovation that happened in the companies I was fortunate to work in, whether it be Bayer, Johnson & Johnson, or Genentech. During my time there, we registered several exciting molecules as new medicines for patients with higher medical need across those three organizations in the 15 years I was there. I think when you move from large pharma to small biotech, I think the focus becomes a lot more laser. So whereas you have the opportunity in large pharma to see several or maybe a dozen different molecules at different stages of development, and you may be fortunate enough to touch on or have accountabilities for some or all of those. When you're moving into the small biotech space, it's usually one or two that are really material to the company. So single and double asset companies are quite common in the biotech space, and very different in terms of the approach which we take in biotech, which is to be very laser focused, not only on the spend, but also in terms of the breadth of your responsibilities. So where there may be a team of 10 in large pharma, there may be a team of one and you're it, which is both fun, but it's also in some stages it can be rather intimidating coming into a new therapeutic area for the first time. What I've really appreciated, though, moving around through my various different therapeutic areas and bringing different molecules to late stage and potentially into commercial, is there's a lot of people out there that can help you with regards to information and guidance and knowledge, those being our scientists, our KOLs, our investigators, and our potential prescribers in the future. And I've been fortunate enough to meet many of those and be humble enough to say, I need your help. I need to learn. And they are incredible sources of information that help me to bring that knowledge back into a biotech such as Jasper to advance our clinical development program.
And of course, speaking of Jasper, we'd love to learn more about the missions and goals of the company and really the focus on mast cell diseases. And if you can tell us more about what those diseases entail specifically, and again, more about what the company is leading in that space.
Yeah, thanks. So our mission is to really address mast cell mediated diseases. Those are, I would say, several dozen mast cell mediated diseases. Our lead asset is briquilimab, which is a monoclonal antibody against KIT. KIT is expressed on mast cells and has really a sole survival pathway. If you block KIT for long enough of its stem cell factor, you essentially starve the mast cell of a critical internal cascade of biochemistry that results in apoptosis. That's essentially the controlled cell death of the mast cell. So we began our clinical development program looking at urticaria, where we know that the mast cell is the central immunological cell in urticaria that leads to hives and itch in many patients. And we think these numbers are maybe around 1% of the population in the United States and in the G7. So that is a very large opportunity to help patients who really have only had antihistamines and omalizumab, which is the available biologic in the past. So this is an antibody that can really drive down mast cells very effectively for these patients. And the key therefore, as in urticaria, was to safely remove the mast cell from the patients for a period of time, and then assess when these patients needed dosing again, when the symptoms start to come back. And so our goal at Jasper is to really bring briquilimab to late-stage clinical trials and registrational studies. And then, if we're fortunate enough from that to show strong efficacy and safety, which we've seen in our early trials, then we would look to register that product both in the United States and globally.
Wonderful. And can you tell us about some of the specific indications that you're pursuing the lead candidate in? And I guess also what the current unmet needs, as we talk about in a lot of these diseases, what would they be in some of the indications that you're pursuing?
Yeah, so our lead indication is CSU, that's chronic spontaneous urticaria. These are patients who have sustained hives and itch, which really haven't got another cause. So there's an inducible form, which we're also examining. I'll come to that in a moment. But if you cannot find a cause for the urticaria, then we term this as chronic spontaneous urticaria. I mentioned that these patients have hives and itch. And typically, these patients are more female than males, about a 75-25 ratio. And typically occurs in adulthood between the ages of 40, but obviously there's populations younger and also older as well. So we are seeing those patients who are refractory to antihistamines. Antihistamine has been a way of controlling the results of mast cell degranulation in the past. And really, those are for I would say, the milder diseases where we are developing our monoclonal antibodies for those patients with moderate to severe disease. And this is measured on what's called a UAS-7 score. And those patients have a UAS-7 of 16 to 42. And what that means is every day they score their hives 0 to 3, 3 being the worst. And so that is when they have unbearable bearable itch. And then also their hives as well. How many hives have they got across their body? And this is a disease which can generally affect the trunk with these sort of nettle sting look at what's called a geographic inflammation of the skin. The hives stay around for about 24 hours and then they appear somewhere else. And so we've been looking for triggers as to why the mast cell misbehaves in this disease so much. And many careers have been spent trying to really establish what are the causes of this mast cell-driven disease. And we see there's two major subtypes, an autoallergic subtype and an autoimmune subtype. The good thing about briquilimab is actually it's agnostic of both of those subtypes, because taking away the mast cell for a period of time, which you can do quite safely, mast cells are essentially a very primitive immunological cell that in really developed cultures today, we don't really need it, because this was as I said, a primitive immunological cell that was typically to respond to venoms and poisons, which we don't typically see in westernized societies today. So we know that we can take away that mast cell for a period of time. And the biology of mast cells means that they have to repopulate into the skin. And so we can really dose our product, our monoclonal antibody, in accordance with when those mast cells start to come back into the skin. What does that mean? It actually means that we may be able to give a drug which drives mast cells down very quickly with within 2 weeks, patients are seeing significant improvements in their hives and itch and relieving their disease, and then looking for that durability before then redosing. And we have some early data to suggest that we may be able to dose out to 8 weeks with our product. What that would mean is a 6 times a year subcutaneous dose of briquilimab, where you have very high control of the disease. This would be very exciting for patients whose patients' journeys have been really quite poor, I would say, over the course of time, that their quality of life has been significantly impaired. And if you could imagine yourself with hives and itch at night, where it's keeping you awake, where you're having poor sleep quality, difficulty with the relationships, even acquiring relationships, may even affect your work by either the itch or the visual rashes that perhaps your colleagues may see. And so having a drug which can help these patients, I think is going to advance and enhance the quality of life of those patients with CSU. Our second indication is the inducible type of chronic urticaria, and we've looked at 2 types, symptomatic dermographism and cold urticaria. So symptomatic dermographism is where there's contact with the skin, which leaves a wheal, which is, again, the mast cell degranulating, where people's clothes touch the skin, or you can actually induce it with just a very light touch on the skin. Again, it's quite prevalent. CIndU is about a third of the total chronic urticaria population, and so it is very prevalent in all countries. The other type is cold urticaria, chronic cold urticaria. This is where patients, if they change their temperature, again, it results in the hives and itch. This sounds quite trivial until you think about younger patients who may be jumping into cold swimming pools, and this can actually precipitate an anaphylactic reaction, which can actually lead to a fatal outcome. So these are not trivial diseases which we're dealing with. Most people have thought over time that urticaria was just, something a little bit more trivial, but we're actually seeing that the quality of life in both types of urticarias can be very significantly impaired. And then also as well, you have that mortality risk associated with cold urticaria as well. So those are the 2 indications in the urticaria space. There is an opportunity for another KIT inhibitor that is exploring atopic dermatitis, and we've seen results from a prurigo study as well, which encourages again in the dermatological space. And then we decided last year that we wanted to explore asthma as our next indication, and we feel that the mast cell plays a very important part in asthma and a number of endotypes of asthma. We are testing in our proof of concept study allergic asthma. And for years, if you've looked at sort of asthma presentations, there's often communication around interleukins like IL-4 and IL-13, and there's alarmins, and there's IL-33 and IL-5, and these are all successful biologics. But what you see on many of the slides at these scientific conferences, you see this little round object called a mast cell. Nobody talks about it because nobody knows what to do with it, because we've never had a tool that can deplete mast cells until briquilimab was available to patients with asthma in our trial. And so what I'm excited about the study, we call it the Atesian study. The Atesian study is going to read later this year. And there, I'm really excited about 2 questions. First question is, for the first time, we're going to know really what does the mast cell play in the role of respiratory disease? And it's in an allergic asthma group of patients that will be depleting the mast cell. And so we want to see, if we take the mast cell out, what actually happens to their disease in terms of an asthmatic response to allergen. And then the second is, do I have a therapeutic? So can I improve the FEV1s and the quality of life of these patients by successfully removing the mast cell for a period of time? We'll have those answers later this year. Very excited about that. And I think that showing our read-through from the CIndU study last year to the Beacon study earlier this year, we know that we can reduce mast cells. And I think there's a promising read-through for these trials, that how the mast cell plays in these very different diseases.
Indeed. And really, I think at the center of this all is really understanding mast cell disease biology. And like you said, there was no way of depleting them or of really getting to know how those targeting them would lead to a clinical benefit. And so now, how do you see our understanding of mast cell biology have evolved, and what are the things that have allowed for that in terms of do we have a better understanding of the markers of mast cells, biomarkers? How do all of those things play into this? And also related to that, really targeting c-Kit here as a personalized kind of or targeted approach, I would say, in terms of identifying this key marker that you can utilize to really target the diseases that you're focused on. So how has the understanding of mast cell biology evolved, and what has been key? Biomarkers, cell surface markers, and all of those things.
Yeah, no, thank you for the question. So I think firstly, the basics are in our early trials, our healthy volunteer studies, we actually correlated the number of mast cells in the healthy volunteer skin as we ascended our doses to understand how much drug you need to deplete mast cells. And there is a threshold. So the mast cell has many, many different receptors, as you'd imagine, because it's one of the early warning signals for essentially an insult or a venom or a virus, etc. And so this thing is armed to the hilt with these receptors, but it also has a number of c-Kit receptors on there, or KIT receptors. And so we need to block as many of those as possible to starve the mast cell of its signal that leads to that apoptosis. And so when a mast cell is activated, it results in degranulation, and that's when the immune mediators such as prostaglandins and histamine and chymase are released, and that's what causes the inflammation in the local tissues, be it in the lungs, in the gut, or in the skin. And obviously in the skin, it's most visual because you can actually see those hives, and patients complain of itch. And that's really the interplay with the nociceptors in the skin or the dermal nerves. There is crosstalk between the mast cell and the nerves. And we've learned quite a lot about that, is that substance P coming from the dermal neurons can actually activate further the mast cells. And so you get into this sort of forward loop cycle that happens. Now, if you take the mast cell out for a period of time, there is the opportunity to really calm all of that inflammation down. Now, with regards to biomarkers, one of the things a mast cell does in general, and mast cells are pretty much all the way around your body, but there are large repositories of mast cells, the skin, the lung, the uterus, the colon in particular. Now, there is an expression of tryptase, serum tryptase, which is expressed basally by all of the mast cells to a greater or less extent. So we can measure a biomarker of serum tryptase, which is a measure of mast cell activity and also mast cell population. So for instance, if somebody has an anaphylaxis, you can measure tryptase because there's a lot of degranulation, and so that tryptase increases in the serum, and then you can show anaphylaxis. Now, in the basal sort of status, the level of tryptase is usually below 8.5 nanograms per mil. And what we found is when we deplete mast cells effectively, we take that tryptase all the way down to zero. And so what that tells us then is that our drug is actually depleting mast cells. And then what we can see then in terms of the duration or the durability of that reduction is understanding when those mast cells are coming back. And this is really the thesis behind when do we really need to redose. And some of the work that we've done here at Jasper in our research lab has actually shown that mast cells don't come back into those different tissues at the same periodicity or the same rate. So there seems to be a preference that mast cells migrate originally or initially to the colon and then start to replenish in the lung and then in the skin. So this gives us ideas and insights into just when we would need to redose a patient who has either respiratory disease or has dermatological disease. So that's a really important biomarker for us.
Wow, that's, absolutely. I think right now the landscape is really asking for those biomarkers to really be able to measure and monitor disease, therapeutic efficacies, and all of those things. And in terms of, since you're looking at different indications here, have there been any cross-disease insights or learnings from asthma, allergy, autoimmune research that are influencing how you're approaching urticaria and other potential programs as well?
Yeah. So these patients, some patients don't just have one particular disease. They may have a constellation of them, and so a patient with urticaria may also have asthmatic disease. So there may be an opportunity for us to gain insight in those patients whose primary driver to come into the controlled study is their urticaria, but we can also understand a little bit more about their asthma as well. This is more, I would say, around the anecdotal side, but we are acquiring data from our clinical studies, which can really cross-pollinate the two programs. We can also learn about GI symptoms as well. So I think in your intro, you mentioned that irritable bowel syndrome, again, is likely to be a mast cell-mediated disease. And again, we can understand the effects of briquilimab in depleting those mast cells, is how that improves the lives of those patients who may have both urticaria and irritable bowel syndrome at the same time. So I think there is some crosstalk. There's an enormous amount of academic work. Steve Garley's lab in Stanford has been hugely insightful in terms of mast cell biology. And then also the charity organization. And I'm sad to say Marcus Maurer, who unfortunately left us in a tragic accident last year, was really a powerhouse for mast cell biology and science, and created the charity organization enormous amounts of scientific collaborations to give us more insights into what exactly is happening with mast cells with these various different therapeutics applied. We miss Marcus a lot, and we're glad that we have Martin Metz and the rest of Marcus's colleagues at the charity. And then enormous amount of work done here in North America as well, in several centers studying mast cell biology.
It's wonderful, and I'm sorry to hear about the loss. And but yeah, it seems like really at the core is at the foundation is having a great team and great partnerships and collaborations. And of course, patient advocacy groups have been a key stakeholder across a number of different areas, especially in rare diseases. And so, can you talk a bit about some of the partnerships and collaborations that have been, and as you mentioned, one of them, and are continuing to be important in driving the company's work forward?
Yeah, so we as a small biotech, we are liaising with the patient advocacy groups. Predominantly, that has been through the charity organization in Berlin, but we're now starting to see emergence of patient advocacy groups in urticaria itself. So the global allergy and asthma patient advocacy group led by Tanya Winders is really starting to expand and have a greater presence, and really representing on a global level patients with urticaria. And so it's wonderful to have that partnership. These are early days for us as a small biotech, just moving now into our registrational programs. But we certainly look forward to having more input from patient advocacy groups and patient representatives for our clinical studies, and how we can be a good partner in society to really help the communities understand what is potentially on offer from our clinical studies and the potential benefits of briquilimab.
Yep, indeed. And in terms of, the broader spectrum of chronic inflammatory or allergic conditions, where do you think innovation is most urgently needed at the moment?
I mean, as an industry, there are enormous high unmet medical needs, as we know. The growing aging population leads to neurocognitive decline, and we're obviously very keen to see a very challenging disease, Alzheimer's, which I think is not my area of specialty, but certainly we're seeing enormous growth there. And I think that there's been many attempts to try to treat that disease, but I think to date, we really haven't conquered that. Really, really excited about, obviously, some of the GLP-1s now that are making a very significant difference to weight loss, which I think is a significant public health issue across the world as well. So those, I think, are two very significant chronic diseases, which I think the biotech and the pharmaceutical industry is addressing or is trying to address now. And then obviously it wasn't so long ago that, we had the emergency with COVID, more acute infections. One of my colleagues at Moderna really predicted that, this was eventually going to happen and that we needed to be in a state of preparedness for COVID. And I'm just glad that Moderna and his team there were in a state of preparedness and was able to respond to that very significant pandemic.
Yeah, indeed. And in terms of really, I think one of the other questions here, yes, in terms of the guiding principles as a leader, chief medical officer at a host of companies, what really now is your focus and your strategy at Jasper in terms of the company's clinical development strategy, and where, what I think you did touch on this and mentioned the status of some of the trials, but where are things right now and where are they heading in a couple of years?
Yeah, so we're a public company. We've guided that our CSU program is planned to start in registrational studies in the second half of this year. I'm very excited about that. That will be informed about the data from the data from the Beacon trial, which is our CSU study. We've shared some of that data both at scientific congresses and then also in company webinars. And so we'll be sharing more data later this year as guided. And I'm looking forward to getting the sites up and running for our first patient dosed in our phase 2b 3 operationally adaptive study in the latter part of this year. We will then look to open another study, another phase 3 study, once we have a final dose selection. And then we're really on our way, we hope, to submission for a BLA in the United States and an MAA in Europe, and have this product commercially available to as broad a community as possible, where we think that the patients can benefit the most. On the second program, CIndU, I think that we certainly would see that as a really good opportunity to move into registrational programs following the data from our Spotlight data. That will be shared later this year at a scientific conference. And then Etesian, the Etesian trial in respiratory disease. I'm excited about all our data, but I'm very excited about our Etesian data because I think that could be transformational if it reads positive. So those are our outlooks, I think, at the moment on the 3 indications. We'd love to be able to choose a 4th indication. As I mentioned at the early part of the podcast, there's probably several dozen mast cell mediated diseases we could probably prosecute with briquilimab. It's just a matter of bandwidth and resource to make all of those things happen. But I think there's still an enormous volume of patients out there that potentially could benefit. You mentioned food allergy. It would be, I think, a really interesting indication for younger patients if we were to run an early trial in food allergy in the future. We have made a decision, so this is not an announcement around food allergy. We're going to do our diligence on next indication, but certainly there's an opportunity there. And I think there's a number of other respiratory opportunities where we're studying that in our Jasper lab right now as well. So I think there's more opportunities than we have resources.
And finally, I'd like to ask you, of course, what advice would you give to aspiring clinician scientists or entrepreneurs in industry who want to really drive innovation in unmet, under-addressed therapeutic areas, as the ones that Jasper is tackling?
Again, my sense of humility here is to be curious, to be humble, to be open-minded, to be flexible. And then also as well, we're in an industry of very high attrition. So you mentioned one of my earlier companies that wasn't so successful, despite a great team and a great molecule. Things don't always work out. Now, obviously, I'm very optimistic about Jasper, and I think it has a wonderful team, a great molecule, really good operating model, and I think the trials which we have embarked upon and we have planned are robust. And I think that as a CMO, you have to do. I think you have to have all of those things. It comes down to having a great team. That's really essential. And then having the trust of the patients and the prescribers. That is really key. And the trust of the regulators too. We've spent a lot of time, and I think this is important for a biotech CMO, is spending time with the KOLs and the PIs. It's a fairly pointless exercise if something is going wrong and that's the first time you're reaching out to discuss an issue. It's far better to establish the relationship ahead of time, build the trust, build the understanding, and really go on the journey together with your investigators and your KOLs so they understand and they can guide and redirect when perhaps their experience suggests that you need to move in a different direction. So I'm always very grateful for advice, and there's never enough advice I can get on a daily basis.
And Dr. Tucker, I think with a great team also comes, it speaks to great leadership as well. So it's obvious to me that you definitely embody that. And that's the key to building a great team. Everything great starts from the top. So we thank you so much for your time today. And thank you so much for leading the work that you do in this very important space. And we'll keep our eyes out for the next milestones coming up from Jasper, and definitely interested in keeping up to date on that for ourselves and for our audiences here as well. So once again, thank you so much for your time and really valuable insights.
Thank you, Ayesha. I've really enjoyed it today, and I really appreciate the opportunity here today. And I hope this helps your listeners understanding urticaria, and maybe it pushes the program along just a little bit further. So thank you so much for your time today.
Absolutely. And really bringing awareness to disease I feel like not many have probably heard of, or maybe heard of in different formats or in different contexts, different names. But really for you to be bringing this to us here, and we're so happy we're going to be able to share this. So thank you so much.
Thanks so much. Have a great day.
All right, that's the end of this episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we will see you next week. Bye for now.
To join the Xtalks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.